Good evening. So far we are discussing the cell organelles. So we have finished the cell wall, plasma membrane, nucleus, chloroplast, mitochondria, and now we are going to discuss regarding this endoplasmic reticulum. So the name itself indicating a reticulum which is found in the center, endoplasmic reticulum, a network of reticulum, tiny tubular or sac-like structures with a single membrane suspended in the cytoplasm, dividing intracellular space into two distinct compartments, that is luminal, that is inside the endoplasmic reticulum, and extraluminal, outside the endoplasmic reticulum, that is cytoplasm. Next, they arise from outer nuclear membrane to the plasma membrane as extensions for the synthesis and transport of protein, lipids and other compounds. So they arise starting from the nuclear membrane and extend towards the plasma membrane. The diagram you can see later. So it was first reported by K.R. Porter in 1945. So he has first reported this endoplasmic reticulum. It exists in three forms, namely cisternae, tubules and vesicles. You remember, three: tubules and vesicles. Cisternae are the flattened sac-like structures. They are arranged parallelly and interconnected. So those are called the cisternae. The tubules are tube-like structures, vesicles are vesicular structures. That's all. This endoplasmic reticulum, this endoplasmic reticulum is of two types. One is the rough or granular endoplasmic reticulum. The second one, smooth or agranular endoplasmic reticulum. So when the endoplasmic reticulum having these ribosomes on their outer wall, those are called rough endoplasmic reticulum. When there is no ribosomes attached to the endoplasmic reticulum, that is called smooth or agranular endoplasmic reticulum. So here the structure is given, the endoplasmic reticulum structure is given here. So we can find, as I told you already, cisternae, they are parallelly arranged. These are the vesicles and these are the tubules. And you happen to see here the small granular structures are there. These are called rough endoplasmic reticulum. When no granular structures, they are called the smooth endoplasmic reticulum. Rough endoplasmic reticulum, the extensive and continuous with outer nuclear membrane. They are studded with ribosomes on their outer surface, appears rough and hence called rough endoplasmic reticulum. They are frequently observed in the cells actively engaged in protein synthesis and secretions. Next, the three-dimensional diagram is given. This is the nuclear membrane, as I told you already. This is the nuclear envelope. When there is a continuation of rough endoplasmic reticulum and they are attached with a smooth endoplasmic reticulum. The three-dimensional endoplasmic reticulum diagram is given. Here the ribosomes are attached. The outer wall, you can find small spots are there, those are the ribosomes. So that is rough endoplasmic reticulum. The remaining one, this is called the smooth endoplasmic reticulum, where the ribosomes are not attached. Regarding the smooth endoplasmic reticulum, the ribosomes free part of endoplasmic reticulum, ribosome free. So there is no ribosomes are attached. Rough and smooth types of endoplasmic reticulum are present in the same cell and they are continuous with each other. That is the other diagram we have seen. They are continuous with each other. Here you can find the rough endoplasmic reticulum, again attach it with the smooth endoplasmic reticulum. Regarding the smooth endoplasmic reticulum, this is absent in prokaryotes. Endoplasmic reticulum is absent in prokaryotes. cells and present in eukaryotic cells. It is well developed in cells concerned with protein synthesis. You can find more endoplasmic reticulum are developed. Regarding the functions, endoplasmic reticulum gives mechanical support to the colloidal cytoplasm. Having ribosomes on its surface, the endoplasmic reticulum helps in protein synthesis. And actually polypeptides are packed in the proteins in endoplasmic reticulum. Polypeptides are packed in the endoplasmic reticulum. Next, it acts as a circulatory system for intracellular transport of various substances. So these endoplasmic reticulum, just like a tube-like structures, are connecting with their parts. So it is used as a circulatory system for intracellular transport of various substances. Lipid synthesis takes place in smooth endoplasmic reticulum. Remember, sometimes given questions, lipid synthesis takes place in smooth endoplasmic reticulum. Detoxification is another important function of smooth endoplasmic reticulum. And endoplasmic reticulum is seat of the origin of Golgi complex. So Golgi complex originates from the endoplasmic reticulum. So here the important things you have to learn. The one is the rough endoplasmic reticulum involved in protein synthesis, and the smooth endoplasmic reticulum involved in the lipid synthesis. Another important thing is detoxification is another important function of the smooth endoplasmic reticulum. Next, we will go to the Golgi apparatus or Golgi bodies. So this is generally called the Golgi complex or dictyosomes. These were first observed as a densely stained reticular structures by Camillo Golgi in 1898 and hence named as Golgi bodies. It is found in all eukaryotes. They are absent in prokaryotes, and especially they are abundant. There are more Golgi bodies are found in granular cells because the main function of Golgi bodies is the secretion function, cell secretions. Next, it is morphologically similar in both plants and animals. There is no difference in plants and animals. In plant cells, the dictyosomes are dispersed throughout the cytoplasm. That is the difference of this Golgi bodies is, in plant cells, these dictyosomes, dictyon means net-like dispersed throughout the cytoplasm. Whereas in animals, they are isolated at some places. That is the difference. Next, here you can see the diagram. The Golgi bodies here you can find. These are the Golgi bodies. The structure I will tell you. These are also you can find cisternae. You can find they are having a tubular structures and broad towards the end, narrow towards the center. Broad towards this one, and then a center lumen. Center part is called the lumen part is there, and cisternae we will see regarding these structures, etc. The next one, each dictyosome consists of various types of morphological components. They are cisternae. Just we have seen that tubular structures having a broad towards the broad structures, and they are arranged parallelly. They are called cisternae, and they are tubular structures. They may be vacuolar structures, and they may be vesicular structures, vesicles. So there are 3 to 7 cisternae which are arranged parallelly. The tubes and vesicles are present at peripheral side. Peripheral side, you have tubes and dictyosome is a polarized structure having a convex proximal face and a concave distal maturing face. As I told you already, here you can find a broad here, and here also have a broad. So appears polarized, having a convex proximal side face and a concave center face. We have the formation of end is closer to the nuclear. This is very closer to the nuclear, and this is closer to the this plasma membrane. Center part is close to the plasma membrane. Transition vesicles formed from endoplasmic reticulum fused to form new cisternae at the proximal end. At each end you can find they are attached. The vesicles are attached at the ends. See here. So these are the cisternae having a broad end at the end and your middle narrow, and some vesicles are attached at the end. These are some vesicles attached at the corners. Vesicles are attached. These transition vesicles formed from the endoplasmic reticulum fuse to form a new cisternae at the proximal end. Next, whereas these cisternae at maturing end are lost in the form of secretory vesicles. They used to remove and form as secretory vesicles. The vacuoles are associated with the dictyosomes, are considered, and condensing vacuoles are pre-secretory granules. The vacuoles associated with the dictyosomes are considered, and condensing vacuoles are pre-secretory, and they are rich in acid phosphates. This is a very important thing. They are rich in acid phosphate. Regarding the functions, this is a very important function, that is cell wall formation. So you know, during this cell division, you can find a phragmoplast is formed. This phragmoplast is formed by the microtubules and the spindle fibers, and then on both sides you can have line of this middle lamella by these Golgi bodies. And cell wall materials like pectin, hemicellulose, microfibrils, etc. are synthesized in Golgi complex. And glycoproteins, glycolipids, mucilage, gums are synthesized and packed in the Golgi complex. So another important thing, glycoproteins, glycolipids, mucilage, gums, etc. are synthesized and packed in the Golgi complex. And another thing, I told you, the endoplasmic reticulum gives Golgi bodies, and Golgi bodies give the lysosomes. So lysosomes originate from Golgi complex. Hormones are secreted by Golgi complex. Different types of hormones are secreted in Golgi complex. Regarding the next cell organelle, that is called the lysosomes. Lysosomes, these are enzyme filled small spherical bodies present in the cytoplasm. So these are also called the suicidal bags. They are discovered by D. Dew in 1955. They are surrounded by a single membrane structures. So far we discussed the endoplasmic reticulum, mitochondria, Golgi bodies, cytoplasm, this plasma membrane, they are all by this bilayered members, but whereas here the lysosomes are single membrane structures. Single membrane structures. It is made up of lipoproteinaceous. They are rich in almost all types of hydrolytic enzymes, hydrolases, proteases, and carbohydrases are all found to dissolve this cell sometimes, that I will tell you later. So optimally active acid at 5 pH, capable of digesting all substances. So lysosomes having the capacity of digesting all substances like carbohydrates, lipids, proteins, and nucleic acids. Membrane sac of hydrolytic enzymes digest or breaks down the macromolecules and enzymes. They originate from Golgi complex, as I told you already. So sometimes what happened, they also originated from endoplasmic reticulum. That's it is mentioned. They originate from Golgi bodies, or sometimes they originate from endoplasmic reticulum. So this is the structure of a lysosome. The outer membrane you have, that is the lipoproteinaceous, as I told you already, and inside you can find number of hydrolytic enzymes. Hydrolytic enzymes are found inside the cell or inside the lysosome. Regarding the functions, lysosomes help in the intracellular or extracellular digestion. In extreme conditions, when the cell is about to die, the lysosomes digest various cell organelles and cell components. that is called autophagy, self-sacrificing. Autophagy, that is, it completely digests all the organelles in the cell. That's all. Next, we'll go to the vacuoles. These are called the storehouses of the cell. Storehouses of the cell appear to be empty but contains cell sap. Generally, if you happen to see in a slide, you can find the vacuole is empty, but actually it is not empty. It contains much amount of cell sap, bounded by a single membrane lashed to the lysosomes, and here also these vacuoles also having single membrane. The membrane of the vacuole is called tonoplast. Remember, this is also a very important point. Sometimes you may be asked. So the outer layer of the vacuole is called the tonoplast. It contains water, sap, excretory products, and toxic products are also found in the vacuoles. Present in all plant cells, but in meristematic cells they are minute, very small, whereas in mature cells they are large and found at the center. But in animals you can find they are very minute or sometimes absent. But vacuoles are present in plant cells. So in meristematic cells they are minute, whereas in mature cell you can find a large vacuole in the center. See, look here, as I told you here, in the meristematic cells, here you can find the central nucleus and having very minute vacuoles here and there, whereas in a mature cell you can find a large central vacuole, and whereas the remaining floral parts are pushed aside. So you can see this is the vacuole, and whereas this nucleus, mitochondria, ribosomes, etc. are pushed aside. Regarding the functions, tonoplast facilitates the transport of a number of ions and other materials against concentration gradient into the vacuole, resulting in high concentration of sap in vacuoles than in the cytoplasm. And the next one is the prevention of cell bursting. Due to increase in the pressure within the vacuole, cell increases its size and swells, but do not burst or break into a certain limit. So this is another function. The first one function is this transport of number of ions and other materials, and the second one, prevention of cell bursting by this vacuole. Imparting colors to parts of plant due to the pigment in vacuoles. So in the vacuoles you can find the pigments like anthocyanin. Here you can find C. Anthocyanins are there, they give color producing, having different colors. Plants due to this important by these vacuoles. And maintain homeostasis. And another thing is the defense, containing waste compounds that are toxic or unpalatable to the herbivores. They may be deposited in the vacuoles. And food vacuoles are formed by engulfing the food. There are different types of vacuoles. Food vacuoles formed by engulfing the food particles from outside the cells, digested them by lysosomes, enzymes by fusion. So this is the food material in the lower animals. They used to take the food engulfing and formation of a vacuole, that is called food vacuoles. The second one is contractile vacuoles. Contractile vacuoles you can find both in animals and also just in Chlamydomonas you can find a contractile vacuole. They used to store the water and burst. So osmoregulation takes place by these vacuoles. Pumps excess water out of the cell by contraction to maintain a suitable contraction of ions and molecules in the cell. So in osmoregulation also they will do these vacuoles. Next, the central vacuoles, they are large, central, derived from fusion of many smaller ones. So very small vacuoles join together to form a very big central vacuole. As there is no excretory system in plants, various types of metabolites, reserve food materials, waste products are dumped in the vacuoles. So These are called the storehouses or repositories of the cell. So vacuoles help in the osmoregulation and food vacuoles, and finally they help in the storing of the waste materials in their cells. Okay, thank you. So we will deal tomorrow some other organelles. Thank you.